Hello, everyone, and welcome to our 27th lecture in our course on brain and behavior. And this lecture isn't going to provide any new content. This is just our review for the third exam. So we'll go over what are the really important things to focus on from the last few weeks of material. So no real surprises here. The structure of this exam isn't going to be any different from the previous exams. You'll have multiple choice questions. As usual, you won't need a Scantron. You just pick the best option among four for the question that's being asked. There will be fill in the blank and matching questions. Those will be a significant part of the exam. For the fill in the blank, she'll be given a word bank, so don't worry about generating your own responses. Do use the word bank. Use each response no more than once. But don't come up with something that isn't in the word bank. There will be some short answer questions. About a fifth of the exam is short answer. So there you just come up with your own response in your own words for whatever the question is. Diagrams will be on the exam, a combination of sort of fill in the label and explain the graph sorts of questions. And then finally, the essay, which has always been part of the exams. This one will cover our discussion of Winston et al., which pertain to our unit on emotions. For which units are on there, it's everything since the previous exam. And this looks like a long list, I know. It's mainly because a lot of the titles for the lectures had ampersands in them, covered a couple different things. It's really no more content that was on the first exam. So emotions, hormones and sex, learning and memory, attention, consciousness and executive function, schizophrenia. We had some smaller topics, so that's why it looks like there are more lectures. We had a lot of single lecture topics, but in terms of information, it's not any more than usual. Before we get going on the review, a couple of reminders. For finals week, be sure to take care of yourself, eat well, get some sleep. We've already seen in our course material that sleep is important. It's important for consolidating memory. It's important for combating stress. You got to stay healthy, so you got to eat well also. And don't cram. Don't wait to the last minute. We saw in our unit on learning and memory that repeated study sessions are the best way to absorb the material. So don't try to absorb it all at once. Don't test yourself just once. Repeated cycles of study and testing are important. And I know that final exams can be stressful. Exams in general can be stressful. But remember that short-term stress response exists for a reason. Sure, your heart rate goes up. You might feel that sympathetic nervous system kick in. But that's there to heighten your senses, to hone your attention, to give you some energy while you go through this stressful event. So remember that short-term stress is okay and even beneficial. A quick word on the extra credit assignments. Make sure that I have those by the beginning of the final exam. So I don't accept late work after that. Grades are due pretty soon after the final. So I don't have a way of updating grades with late extra credit assignments. So if you're going to do extra credit, be sure I have that by the beginning of the test. For our unit on emotion, know the theories of emotion. We have our folk psychology notion, which is that some event out in the world is processed by our brain. We feel an emotion as a result of that stimulus, and that our physiology, our autonomic systems, respond to that emotional experience. In other words, we feel afraid, and then our heart rate increases, or we get clammy skin. That those physiological responses are the result of the emotional experience. In contrast, we have the James Lange theory, where an event causes interpretation, but then what happens first is the physiological arousal. Your heart rate goes up, your pupils dilate, and that afterwards your brain interprets those physiological responses and generates the emotional experience. So in other words, you have a stimulus, your heart rate goes up, and then your brain interprets that, and then you feel afraid. We know that neither of those is exactly right. There's the Cannon-Bard theory, which says these are separate processes, that they occur simultaneously, or at least one doesn't depend on the other, which gets around problems like when you're embarrassed, you feel embarrassed first and then blush, whereas when you are afraid of something, you might have a sympathetic response and then the emotion. So this gets around that, but we know it's not right either. Thanks to the Schachter-Singer experiment, we know that these things interact. And of course, at a very basic level, know what emotions are. So be able to identify the properties of an emotion. And know some of the experiments. Know in particular the fear conditioning experiment. So we have a rat that at first doesn't respond much to a tone when it comes on. It might freeze a little bit, but then it associates the tone with a shock because those are presented together over and over again. And then after that, all it takes is the tone to induce a fear response in the rat. So this has to do with emotion. It has to do with fear. It's obviously also part of our learning and memory unit as well. And remember that fear conditioning depends on the amygdala in particular. So when you destroy the amygdala in a rat, it doesn't learn this association. Know the facial feedback experiment as well. So what forcing yourself to wear a certain facial expression, a smile or a frown, what that does to your actual mood. So understand that concept. We spent some time talking about stress. And as I just mentioned, stress isn't all bad. There are some positive effects, especially in the short term. So understand what those positive effects of stress are and why they are beneficial. On the other side, know what the negative effects of long-term stress are and why they are harmful. And a lot of this is just the other side of the same coin. So you want to, for example, suppress your immune response in the short term so that that energy is available to feed your muscles. You want to get away from the threat or combat the threat. So short-term loss of immunity is beneficial. It frees up energy. Long-term, it makes you prone to disease. So that is obviously a negative effect. So understand all of those trade-offs, not just the immune response part, but understand what the positive and negative effects are of short-term and long-term stress, respectively. Understand our unit on the hormones. So understand what hormones are, how they were discovered in the very first place. We remember Arnold Berthold's experiment on roosters, where the testes were removed from the rooster, and then one was implanted back in the abdomen. And what happened to the rooster's development? We already knew how a rooster normally develops. We know what happens if you remove the testes. And what Berthold's contribution really was the implantation part of this experiment. So what happened in that experiment? What did that tell us about what hormones were? So what were the implications of the experiment? This is, again, a long time ago. It's how we discovered hormones and how hormones were not neural signals. They were more global chemical signals. Also, when it comes to roosters, for example, and chickens in general, understand what testosterone does. So we know, for example, that testosterone is produced by both males and females, but that testosterone seems to be required for some aggressive behaviors, sexual behaviors for the males, the roosters. But it's not the case that more testosterone means more aggression or more sexual behavior. That seems to be an individual difference between roosters. That's not just how much testosterone is floating around. So understand that relationship. For hormones, know what the targets are for these chemicals. And of course, we contrasted hormones with things like neurotransmitters, which have very specific targets, very local targets, just the next neuron. Hormones can have their effect on tissues throughout the body, so a much broader set of signals. Pheromones are chemicals that affect behavior of another member of the same species. So obviously it's affecting tissue in another organism. So that's an even broader signal. And then, of course, allomones affect other species, not one's own species. So we see sort of a trend of decreasing specificity of the signal, what it affects. So understand these sort of cycles, these feedback loops within hormones. So what hormones do within the body. We'll restrict it to hormones now. So know what a releasing hormone is. The hypothalamus secretes the releasing hormone. It makes its way to the anterior pituitary, and the anterior pituitary responds by secreting tropic hormones. So know what those terms mean. Chemically, know what hormones are. So we have peptide hormones, amine hormones, steroid hormones. We know that the peptide hormones are the biggest, physically. We know that the steroid hormones can pass right through cell membranes. So be aware of that. Know how hormones are regulated. So what controls their levels? And of course, the biggest factor here is the idea of negative feedback. When those tropic hormones reach endocrine cells, they secrete their own hormones, and those hormones inhibit the hypothalamus and the anterior pituitary. It keeps them from releasing more hormone. So there's that negative feedback concept. So understand what that means. For the anatomy of the endocrine system, we have the anterior and the posterior pituitary, so know the difference. Know that the structure we're seeing here detailed on the right is the anterior pituitary, and it's governed by the hypothalamus, part of the brain. And the hypothalamus, of course, sends neuroendocrine cells down to blood vessels. Neuroendocrine cells look like neurons, but release hormones into the blood supply directly. They do not signal nearby neurons. So know what those terms are and know how they relate to the anatomy of the endocrine system. Moving on to learning and memory. So we have this diagram, this hierarchy of long-term memory. Be sure to understand this diagram. Know which parts of the brain are important for each kind of memory. So know that episodic and semantic belong to declarative, but also know that the hippocampus is very important for declarative memory. The cerebellum is important for conditioning. The basal ganglia is important for skill learning. We've already seen that the amygdala is important for fear conditioning in particular. So know the kinds of information or behaviors. Know what skill learning is. Know what semantic memory is. What sort of information is acquired. And know the processes of memory. And what I mean by that is the difference between encoding, consolidation, and retrieval. Know how each of these processes relates to the movement of information from sensory memory, short-term memory, long-term memory. So know what that process means. And know how memory can exhibit dysfunction. We can lose parts of our memory. The most famous example, of course, is patient HM, who lost both hippocampi and could no longer form new declarative memories. He still had his old declarative memories, and he could still learn skills. He could still be conditioned, so he didn't lose everything. He just lost the ability to form new declarative memories. And the sorts of things he could learn told us a lot about what the hippocampus does and what it doesn't do, and what the structure of memory is. And of course, HM had a form of anterograde amnesia. So know the difference between retrograde and anterograde amnesia, what those terms mean. And for HM, again, know what kinds of memory he kept, which kinds he lost. What skills, what forms of learning did he keep or lose as a result of that? Here we see the mirror tracing task, which HM could still do because it's a skill. It is non-declarative, whereas he could not remember events in his life and couldn't learn new words because those are declarative memories. And know the experiment and what kinds of memories they test. So mirror tracing, we just saw, is one of them. Episodic memory, we know we use the Ray complex figure test because it's something you have to remember that doesn't have any meaning. The image you see on the right cannot be described in terms of meaning. It's not as though it's a picture of a house. It looks kind of like a house, but if you just remember, oh, it's a house, you're going to miss a lot of the detail. You're not going to be able to recreate that figure very well. And so that's why the Ray complex figure test tests episodic memory. All you have to go on is your recollection of events, what the experience was like. And that's in contrast to semantic memory, where you use the pyramid and palm tree test. The person may not have experienced being at the pyramids. In fact, you wouldn't use the test if they had. But the person just has to know semantically that palm trees are associated with desert environments, and therefore the pyramids. Spatial memory we talked a lot about. We have the Morris water maze, where a rat has to navigate that water-filled tank trying to find that platform. So understand how that experiment works, how Tolman's cognitive map experiment works, where Tolman gave the rat the chance to learn where food was and tested whether the rat was just learning a sequence of turns or was actually learning more abstractly where the reward was. And it turns out it was the latter. And again, skill learning like mirror tracing, like reading text backwards, know that those relate to skill learning and the basal ganglia. Other forms of memory and learning, habituation. So understand the aplesia experiment, that sea slug, or if you poke that siphon over and over again. At first, aplesia retracts its gill to keep it from getting damaged. After a while, it gets used to that stimulus and stops retracting it. And people do the same thing. They get used to a stimulus. They'll stop responding to it. So that's habituation. Understand that experiment. Classical conditioning, an important concept in learning and memory. So know what the unconditioned stimulus is, what the conditioned stimulus is. Know the unconditioned and conditioned responses in, for example, the Pavlov's dog experiment, or the eye blink conditioning experiment with the rabbit, or fear conditioning. Those are all forms of conditioning, and they all have a US, a CS, a UR, and a CR. So understand what those mean and be able to identify them in an experiment. Again, fear conditioning, I don't have the slide up here, the image up here on the slide. It's also part of our unit on emotion, but know how it relates to classical conditioning as well. Know the difference between short-term and long-term memory. The serial position effect, which includes the recency and primacy effects. Recency effect, you remember the most recent words in the list because they're still in short-term memory. The primacy effect, you remember the first words in the list because they've had time to make it into long-term memory. So understand that distinction and which of those effects depend on which form of memory. Smaller scale, the neural mechanisms of learning, how we change the strength of a synapse, what the strength of a synapse means. So that process depends, we think, on long-term potentiation. So understand the steps of long-term potentiation. Know what long-term potentiation is, but know what the steps are. It's a series of cause and effect. So we know that those NMDA receptors are plugged by magnesium. Normally, glutamate alone doesn't let them open. So nothing happens for NMDA receptors. Glutamate does cause those AMPA receptors to open, and that lets sodium in, depolarizes the neuron. And that depolarization, when combined with the presence of glutamate, is enough to pop out the magnesium plug, and now calcium flows in through the NMDA receptor and sets off that second messenger cascade. And now that chain of chemical reactions causes the production of more AMPA receptors, and now that postsynaptic neuron is more sensitive to glutamate. It has a bigger response. The synapse has been potentiated. So understand that process. Also, the small scale, we talked about dopamine. And we know that dopamine seems to encode predictions about reward in particular. So when something better than expected happens, we get a dopamine burst. We weren't expecting that reward and we got one. If something worse than expected happens, we get a lapse in dopamine. We see a dip in dopamine release because we didn't get the reward we expected. So understand that dopamine is also involved in learning. It's not just glutamate. Dopamine is involved in particular in learning about rewards and predicting rewards. Moving on to attention. Know the elements of attention. We know that attention has a limited capacity. You can't pay attention to everything. We know that it takes effort. So you have to try to pay attention. It takes energy. We know also, especially from experiments, that we ignore the irrelevant stuff. We focus on what we're paying attention to, but it means we might miss something that we're not paying attention to. However, the bonus, the pro of paying attention, is that you're more likely to be accurate or make a better decision about the thing that you are paying attention to. So those are the sort of the things that attention involves, what attention does. And we know that we have different kinds. We have overt and covert attention. The difference between looking at what you're paying attention to and looking at something other than what you're paying attention to. Endogenous and exogenous attention. Understand that distinction. So endogenous attention is under our control. It's top down. It's where we decide what we pay attention to. Exogenous attention is driven by the environment. When something surprising happens, a bright flash, a loud noise, that captures our exogenous attention. And for at least a little while, we're going to pay attention to the source of that stimulus. Know how these things relate to the brain. We have the frontoparietal network and the temporoparietal network. those seem to relate to endogenous and exogenous attention respectively. So understand that relationship. And there are a number of interesting experiments on attention. So there's the invisible gorilla experiment on inattentional blindness, where participants that were paying attention to a very hard ball passing task missed the person in the gorilla suit. Because they're ignoring the irrelevant stuff, they didn't notice the person in the gorilla suit. They were focused on their task. The auditory N1 effect. So we see a brain-based result of attention. We see a boost in neural activity detected by EEG when someone is paying attention to a stimulus, even if it's the same stimulus and they're not doing anything else. We see an increase in neural activity. So we know that attention itself has an effect on the brain. Know the difference between a feature and a conjunction search. So feature searches, you're just picking out one feature. It pops out right away. It doesn't matter how many stimuli there are on the screen. Conjunction search, you're looking for a particular item that has multiple traits. You have to go item by item, so the number of items matters. And there are also patients that have attention deficits. So hemineglect, where the person can't pay attention to half of their visual world. It's not that they can't see it, it's that they can't focus on it. And balance syndrome, where someone develops simultagnosia. You get hyper-focused. You can't pay attention to more than one thing at once. So in this experiment, if you have to compare two different objects, someone with balance syndrome can't do it because they can only focus on one thing at a time. So understand those disorders of attention. Consciousness. So there's been some research on consciousness. It's first be able to define it. What is it? Know the difference between the easy problem of consciousness, where we can tell what someone is experiencing based on comparing their brain activity to previous experiences, so identifying the stimuli they're getting. And that's in contrast to the hard problem, where we actually know what that experience is like. We feel what they're feeling, or we know what the color red looks like to them. We haven't solved that one yet. We may never solve it. But that's the difference between the easy problem and the hard problem. Identifying someone's experience versus knowing what that experience is actually like. And of course, what those experiences are like is the idea of qualia, those things that are subjective and are inherent to the experience but can't be described. You can't describe what red seems like to you. You can't describe what pain feels like to you, really, and have someone know exactly what you're feeling. So those are qualia. And part of that same lecture was executive function. So again, be able to define what executive function is. But we talked about several tasks that measure it. So there's the oddball task, where you have to override a rule that you've learned in order to apply the more difficult but more accurate rule. So understand what the oddball task is, be able to describe it. The Stroop task, be able to describe what the Stroop task is. Why is it difficult? What mistakes do people make and why? What is it about executive function that pertains to the Stroop task? The go, no-go task. This also applies to our unit on schizophrenia, but that's where you develop a habit of pushing the button after most of the letters. Every now and again, there's a letter you're not supposed to push the button afterwards, and yet you might make a mistake. That is the no-go trial, and yet you might press the button anyway because you're so used to it. So that's where we have to inhibit habitual behavior. Our schizophrenia unit. So know what schizophrenia is in terms of symptoms. We have positive symptoms. Know what the term positive symptoms means, but also know what the positive symptoms are and what those terms mean. Same thing for negative symptoms. Know what a negative symptom is, but also know what the negative symptoms are. Cognitive symptoms, same thing. Identify what cognitive symptom means, but know what those cognitive symptoms are. Down here at the bottom we have, for example, an inability to pursue an object with smooth eye movements. Know the risk factors for schizophrenia. We know that there are genetic risk factors as well as environmental ones. Know what those are. Know that there's a big component of each. Know what some of those environmental risk factors are. We know that urban environments are a risk factor. We know that stress early in life, prenatal development, and risk of disease are environmental factors that affect schizophrenia. And know how these things interact. So we see here in the bottom left, that familiar graph where on the x-axis you have genes that favor schizophrenia, on the y-axis you have environmental stressors, and these two combine. If you have a lot of both, you will likely develop schizophrenia. If you have a lot of one and not many of the other, you can still develop schizophrenia or something like schizophrenia. If you don't have many of either, you're probably not going to develop it. So understand that interaction. Understand the relationship of schizophrenia to the brain. So there are structural changes that we've seen. Bigger ventricles in individuals with schizophrenia. Neural loss, so the loss of, in particular, frontal and parietal cortices, also temporal lobes. So we lose neural volume. Part of that is the loss of dendritic spines. So individuals with schizophrenia have fewer spines on their dendrites. That means fewer synapses, also means less overall volume. We know also that neurons in the hippocampus, for example, are disorganized. They don't all point in the same direction like other neurons, typical neurons in the hippocampus do. And what causes schizophrenia neurally? Know what the dopamine hypothesis is, what it says about dopamine levels, why it's not complete, it's not a total description of schizophrenia. Same thing for the glutamate hypothesis. Understand what it is, how it relates to drugs like PCP, how dopamine relates to drugs like amphetamines, and how neither of these is the complete story for schizophrenia, but know the evidence for these hypotheses. And then finally, the essay will be based on the Winston et al. Paper. So be able to identify what the experiment was, what the stimuli were, what participants were asked to do in terms of decisions made, what they saw, what sort of judgments they made. Know what the findings are. Know the different parts of the brain that were revealed in this analysis. Which parts of the brain responded to which aspects of the task. We know that, for example, the superior temporal sulcus responded to the explicit versus implicit nature of the judgments. We know that the amygdala responded to whether the face was trustworthy or not. So understand that experiment, understand those results, and understand the implications of the experiment. We talked about many things this experiment tells us about amygdala function, about social judgments. So be able to describe all of that. You can even practice writing little essays. You know what the format is at this point. So that's something you can work on. Okay, with that, that concludes our exam review. So I will say good luck on exam 3, and that will be the end of the course. Have a great break, and I hopefully will see you in a future psychology class. If you are graduating, congratulations.